Hey all, so today we're going to be looking at D3.2, inheritance, and we're going to be talking about the basics of Mendelian genetics. So what we need to be able to do at the end of this video is explain how dominant and recessive alleles can show up in the genotype and phenotype of organisms. So we want to know all of that vocabulary and be able to explain it. So we're looking at page 692 to 699, and our theme is continuity and change. So when we are talking about gametes, first we need to understand that body cells are diploid, and body cells, somatic cells, are body cells, and those would be like skin cells or liver cells or brain cells, and they have two of each chromosome, one from the mom and one from the dad, and those are called homologous chromosomes. And so in all of my 23 pairs of chromosomes, I have chromosome 1, one of them's from mom, one of them's from dad, and these are homologous. And so there are two alleles, one on this chromosome and one on that chromosome. So like if the tip of my finger here were a chromosome, an allele, let's say for blood type, then I would have an allele for blood type here, and I would have an allele for blood type here in the same position on each chromosome. And they're both coding for blood type, but I might have two different ones. So I might have an A allele and I might have an O allele, or I might have an A allele and a B allele. And so alleles are versions of a gene. And so A versus B blood, those are different alleles. Gametes are haploid, meaning they have only half the DNA. And this is achieved through meiosis. This allows gametes to join with another gamete to make a diploid cell. So the only cells in the body that are haploid are egg cells and sperm cells, or reproductive cells, so gametes. Skin cells are diploid, brain cells are diploid, but haploid cells need to be haploid because they're joining together. So they need to have half the DNA so they can join together to have full DNA. Otherwise, you'd have full DNA joining with full DNA, and then you'd have 2 times the DNA. And then you'd have 2 times the DNA and 2 times the DNA going to 4 times the DNA. And so your nucleus would get very rapidly too crowded. So genetic crosses can be used to see this process by carefully pollinating flowers. And you can use like a paintbrush to transfer pollen, and then you keep the stigma in a paper bag to prevent wild pollen from getting on it. And that is exactly what Mendel did with his pea plants. So just before we go on to talk about Mendel here, you can see these homologous chromosomes, one from mom, one from dad. And in this case, they're talking about the allele for free earlobes. So whether or not your earlobe's a bit dangly or it's attached directly onto the side of your head. And so you can see here, the alleles are in the same position on the chromosome. This is called a locus, and they are different. And it looks here when they are documenting this, that the allele for free earlobes, capital E, is the dominant trait, whereas the attached earlobes is the recessive trait. And so this person has both of those alleles, so therefore they would have this trait manifesting in their physical appearance. And we'll talk about this in a minute. So Mendel took pea plants to determine how genes were passed on to the next generation. And he did artificial pollination of all of these, and he was really into pea plants. So P generation is the parent generation. F1 is the first filial generation, which is the first children. And then F2 is the second filial generation. And he counted what traits showed up, what physical traits, and in what ratios. And so Mendel created peas that were pure breeding first. So they only produced dominant or recessive traits. So for example, they only had purple flowers, or they only produced white flowers, and their babies produced white flowers, and their babies' it was only white flowers. And so then he crossed them using these different 7 traits that we have here for pea plants. We're just going to focus on the purple and white flowers and maybe the yellow and green peas. But when he first crossbred them, he didn't see any intermediate traits, so it wasn't like a combo purple-white. It was just that in F1, what he did see was just all purple flowers. And in F2, though, he saw that 25% of the plants were showing this recessive white flower. And so what was going on in F2 is he had these were no longer true breeding plants, in that they would always produce purple flowers. So in F1, they were true breeding. The parent generation was true breeding plants, so they would only produce purple flowers and only produce white flowers. But then if he crossed F1s together, some of them would have the dominant allele, which is the purple flower, and some of them would have the white allele, which was white flowers. And if you then cross the F1 generations, you're going to end up with some of the F2 generations having two alleles for the recessive trait. And that's why they showed up having 25% of the recessive traits. Alleles are created by mutations, and sometimes those are very minor. Sometimes they can be serious. So when we're documenting alleles, we are going to use a capital letter for a dominant trait, and that's going to show up in the phenotype, meaning it shows up in the physical characteristics. And then the recessive is going to be used documenting a lowercase letter, like the small y. And that's going to be hidden if there's a dominant trait, even though that small y will be in the genotype. So the genetic material has that allele, but the dominant trait is blocking the recessive trait from production. So this can be affected, physical traits can be affected by genotype only. So that would be something like your blood type. You either have A blood or you have O blood or you have AB blood, whatever your blood type is, but whether you eat more vegetables or not, that's not going to change your blood type. Your eye color, same kind of thing. And then there's some things that are going to affect your physicality that are just environment. So like I have a tattoo. That is an environmental thing that has nothing to do with my genetics, scars, things like that. If I lost a hand in an accident, those are environment things only. But then there are some physical characteristics and some characteristics in our body that are going to be affected by both. And so height is a good example of this. Genetically, I might have tall person genes, but if I am poorly fed when I'm young, then I will grow less. This is the same with diabetes. So I might be prone to diabetes, but if I eat very healthily and exercise regularly, then it's much less likely that I'm going to get diabetes. So when we are talking about genetic traits, a tool that we use is Punnett grids, or you might call them Punnett squares. And so you can see one here over on the right, and yellow pea color is for pea pods is dominant, whereas green pea pods are recessive. And so when we are crossing one that is dominant here, but it has a combo allele here, combo allele is called heterozygous, meaning they're not the same. And this one is homozygous, but it is homozygous recessive because they are the same, but they are both recessive alleles. So when we have a parent cross like this, the way that we use our Punnett grid is we're going to document the capital Y here and then the small y, capital Y and the small y. And then in this box, small y, and then again here, small y. And so what that means is that the likelihood of this parent cross producing offspring that have yellow pea pods is going to be 50%. And this is a monohybrid cross, meaning we're only looking at one trait, pea pod color. We're not looking at pea pod color and pea pod texture. We're just looking at one trait. And that's what we need to know for SL. HL will look at two traits. So in this case here, we are actually doing a test cross. So we do test crosses because we don't know if this yellow pea pod parent is homozygous dominant, meaning it has two capital Ys, or if it's heterozygous. And so we can do a test cross, see the offspring, and then we can know the genetics of the parent plant. And so in order to do a test cross, though, we have to cross it with the recessive trait. And in this case, from the offspring, we can see that there's 50% showing up as yellow and then 50% showing up as green. And what that means is that this parent is heterozygous. If this parent were homozygous dominant, what we would see is that all of the pea babies, their pea pods would appear yellow. So heterozygous is a dominant and a recessive allele. Homozygous dominant is two dominant alleles, and homozygous recessive is two recessive alleles. And so one thing to keep in mind is that there is phenotypic plasticity in organisms. And what that means is that they change their expression due to the environment, but it can go back. So an example of this would be melanin making pale or less dark skin darker. And so if you've gone through the winter in northern Canada, for example, and you notice that your skin is paler, that means that the melanin is not being produced as much, because melanin is a pigment that protects you from UV light. And so, but in the summertime, you might get a suntan or your skin might become darker. And that's where your skin is making more melanin. And so this gene is being expressed to produce melanin to protect your skin from UV light. And this is called phenotypic plasticity. So when we are talking about alleles, one thing that's going to come up potentially is genetic conditions. And some genetic conditions are dominant. They're rare, but they're possible. And an example of this would be Huntington's disease, which is a neurodegenerative disease. And so what's going on with Huntington's is if your parent has Huntington's, because this is a dominant allele disease, then you would have a 50% chance of having Huntington's. And you, if you have Huntington's, would have a 50% chance of passing it on to your offspring, because you only need one of those dominant alleles in order for you to have Huntington's. So recessive genetic conditions are things like cystic fibrosis, which is where excess mucus is made in the lungs and the digestive tract. It causes you difficulties breathing and then also digesting food. Phenylketonuria, where you can't make the enzyme to convert the amino acid phenylalanine to tyrosine, and this leads to intellectual disabilities. And then there are some diseases that are on the X chromosome. So these are called sex-linked traits. And some of you might have heard of red-green color blindness. So that is on the X chromosome. They're all on the X chromosome. And so what that means is for males, they have an X chromosome and a Y chromosome. And when you are male, you don't have two X alleles. And so if your mother was a carrier for red-green color blindness, then you got that allele from her, then you would be red-green colorblind if you were male. If you got that carrier allele from your mother, but no allele from your dad, that your dad's allele was that he could see red and green, then as a female, you would have an allele that coded for correct eye pigmentation proteins. And so then you would be able to see red and green. So it's rarer for females to then have these genetic conditions because they have two X chromosomes instead of having an X and a Y chromosome. Hemophilia is another sex-linked trait genetic condition. And so this is where an individual would not make clotting factors. And then some are codominant traits. And so an example of this is sickle cell. And so if you have the sickle cell allele and you have the normal red blood cell allele, then you will make both sickles and normal red blood cells. And this is great because it makes you resistant to malaria, and you can still breathe functionally. But if you had both sickle cell alleles, then you would make only sickles, and it would make it very difficult for you to breathe. And yes, you would be resistant to malaria, but oxygen. And if you had both normal red blood cell alleles, then you would make normal red blood cells, but you wouldn't be resistant to malaria. So to summarize, gametes are haploid. That means they're one N, one strand of DNA, like one copy of chromosome 1, one copy of chromosome 2. Alleles are variations on a gene. Mendel is the father of genetics. That's him right there. And since we were just talking about red-green color blindness, this is a circle. For testing for red-green color blindness, there's a number in here in pink, the number 9, and then the number 6 in here in light green. But it would be very difficult to see this if you were red-green colorblind. And then if we know the genotype of an organism, we can predict potential offspring they will create using a Punnett grid. And dominant traits show up in the phenotype. Recessive traits are present in the genotype, but don't show in the phenotype unless they're only recessive traits in the genotype. And then we need to know the terms heterozygous and homozygous. And so homozygous dominant or recessive means 2 dominant traits, 2 recessive traits, whereas heterozygous means 1 allele of each. And so here we can see the inheritance pattern of cystic fibrosis. And so you can see here that both parents are carriers of the recessive allele. And so when we do this Punnett grid, there's a 50% chance that the children would be carriers, but they would not then express that disease. And so they would not be sick. So 75% of the children would not be sick with cystic fibrosis, but 25% of the offspring potentially could be sick. And so because cystic fibrosis is a relatively rare genetic condition, the likelihood of both parents being carriers is rare. You would probably have one parent potentially being a carrier. And so then your children could be carriers, but would be unlikely to have the genetic condition. All right, that is all for this video. Thank you for watching.